adenosine triphosphate, or ANT, is a molecule that plays a crucial role in cellular energy metabolism. It is a nucleotide consisting of an adenine base, a ribose sugar, and 3 phosphate groups, linked by high-energy phosphoanhydride bonds. ATP is synthesized during cellular respiration, a process that occurs in the mitochondria of eukaryotic cells and involves the breakdown of glucose or other molecules to release energy. ATP acts as a universal energy currency in living organisms, providing the energy needed for cellular processes such as muscle contraction, active transport of ions and molecules across cell membranes, and DNA synthesis. It is also involved in signaling pathways and regulation of enzyme activity through phosphorylation reactions. ATP is synthesized through 2 main pathways: oxidative phosphorylation and substrate-level phosphorylation. In oxidative phosphorylation, electrons from glucose or other molecules are transferred to a series of electron carriers in the mitochondria, generating a proton gradient across the inner mitochondrial membrane. This gradient is used by ATP synthase to produce ATP from ADP and inorganic phosphate. In substrate-level phosphorylation, ATP is synthesized directly from ADP and inorganic phosphate during the breakdown of high-energy molecules, such as glucose or glycogen. ATP is constantly being hydrolyzed to ADP and inorganic phosphate by various cellular processes, releasing energy that is used to drive other reactions. The regeneration of ATP from ADP and inorganic phosphate requires the input of energy, which is usually provided by oxidative phosphorylation or substrate-level phosphorylation. ATP is also involved in various regulatory mechanisms in the cell. For example, it can act as a signaling molecule by binding to purinergic receptors on the cell surface, triggering intracellular signaling pathways that regulate cellular responses such as inflammation and pain. It can also act as an allosteric modulator of enzymes, regulating their activity by binding to specific sites on the enzyme molecule. In summary, ATP is a critical molecule in cellular metabolism, providing the energy needed for cellular processes such as muscle contraction, active transport, and DNA synthesis. It is synthesized through oxidative and substrate-level phosphorylation pathways and is constantly being hydrolyzed and regenerated. ATP is also involved in regulatory mechanisms in the cell, acting as a signaling molecule and regulating enzyme activity.